Hi, I'm Kendra, and today I'm going to be talking about meta-analysis decision trees, which will help you choose which meta-analysis method to use in your meta-analysis. First, a little recap from previous presentations. You've learned so far why you want to do a meta-analysis and a little bit about manual versus automated meta-analyses, as well as the difference between coordinate and image-based meta-analyses. And I'll talk a little more about these differences, but if you don't have just a general idea of these points, I suggest you go back to previous lectures and rewatch them. For context, in a figure that you've seen, this is the figure showing the different steps in a meta-analysis, and we're, in a manual meta-analysis. But we're at this stage of the deciding specific analyses and potential sub-analyses. Keep in mind that this is just a guide to get started and you don't need to understand everything. I don't understand everything about every single analysis, every single method. I don't think anyone does, to be honest. But we know enough in order to teach you how to make a good decision, and then you can learn more about the particular method that you're using. So here we have the first decision tree, which shows you how to pick a manual meta-analysis method from Nightmare. Nightmare is a neuroimaging software in Python, and we're focusing on Nightmare because that's what is used for the demo later, and also because it implements the widest variety of methods, so we can talk about many different methods that you can try all with the same software. So the first question we need to ask is, do you have the results images? By results images, I mean when somebody runs a participant level study and analyzes the results, they'll end up with some kind of group level map usually, and this group level map is the results image. So if you have those images, you'd be doing what's called an image-based meta-analysis. Now, it's a little bit more rare to do these because people don't always share their results images, but I'm going to talk about it first because I think it's a little easier to understand the coordinate-based meta-analysis if you have in your head how it would work for an image-based meta-analysis. So we'll start with this side with the image-based meta-analyses. First of all, if you have the standard error images, which is another type of image, but with the error, the variance or the standard error between participants, then you can do a mixed effects general linear model. And there are a few other technical questions that you can ask yourself in order to choose which specific method, but generally it works like this. At the first level, the original authors for each study would be modeling the time series and have a map and a variance map for each participant. At the second level, they'd be modeling these participant level maps with the variance into a second level study level group map. And then at the meta-analysis level, at the third level, that's a lot of levels, you're modeling the maps across different studies along with their error maps in order to get a meta-analysis map with an error map. So this is sort of the gold standard for meta-analysis. This is the best way to do it if you can. If you don't have those error images, the next question is whether you have Z-maps or T-maps. And one question that you ask for either of these answers is whether you have the participant size, the participant sample sizes, meaning the number of participants used in the individual studies. And this is important because you want to give less weight to studies that have very low sample sizes because they're likely noisier and have less reliable results. So that's what the weighted part of this weighted Stouffer's name refers to. You're downweighting studies with very low sample sizes. Now let's talk about the other side of this question, coordinate-based meta-analyses. And the first question you can ask yourself is whether you want to look at activation or functional connectivity. So let's start on the side of activation. The next question is if you want to summarize one group of papers or compare multiple groups. So say I want to do a meta-analysis of autism papers, or I want to do a meta-analysis of, say, autism children versus autism adults. You could use different methods depending on how many groups of papers you have. So if you have one group, this is the most basic coordinate-based meta-analysis, so I'm going to spend a little more time on it. If you have one group, you're going to ask yourself this more technical question, which should make sense in a few slides, of whether you want to use a continuous Gaussian kernel or a flat kernel. So let me just explain what this kernel is. So here on the left we have maps from individual studies. The black dots are the coordinates of peak activation reported in those studies. And then this is a map where those peak coordinates are combined across studies. So then we convolve all these coordinates with either a density kernel or an activation likelihood estimation kernel. So this kernel, the AL kernel, is Gaussian. It's continuous, so it's highest right in the middle where the coordinate is, and then it gradually tapers off to 0. Whereas the density kernel basically just makes this sphere bigger. It's all 1 in the sphere and 0 outside the spheres. So then you get a density map from all the different pooled, convolved maps from individual studies, and then you apply a significance threshold in order to get your final results. So now I'm just going to explain the difference between KDA and MKDA, because I don't know if you noticed on the decision tree that there was another method that was crossed out, that was KDA, because MKDA is an improvement on KDA. But in order to understand that, I'll briefly explain what KDA is. So KDA is kernel density analysis. In the previous figure, in this figure you saw that the density kernel is just binary. So each dot is basically made bigger. So you can imagine this is a brain instead of a square. And these are the coordinates reported. Each dot is made bigger into a bigger circle. So it has the value of 1 in the circle and 0 outside. Where there's overlap, it has the value of 2. And then when you pool across different studies, so this is one study and this is another study, when you pool across many studies, you'll get a map like this. And once you've thresholded it, you might end up with just keeping the areas where you have the number 3 or the number 2, etc. On the other hand, MKDA adds another step. That's why it's called multi-level kernel density analysis, because it adds another level. So this first level is the same. And then whether there's overlap between convolved coordinates within the same study, they binarize the result. So each individual study contributes a binary map. And this means that studies that report many coordinates close together won't be contributing more to the final map. And this has been shown to be a better method. So here you can see the same data meta-analyzed with different kernels. Here's the multi-level kernel density analysis kernel, which is the binary kernel that's binarized within studies. So each study contributes a binary map, like I just showed you. Here's the KDA kernel where they don't do that binarization. So you can see, for example, right here, there's a little area where these two dots overlap, and that area is higher. And then you have the AL kernel, which was the continuous Gaussian kernel, going from the highest point in the middle down slowly to 0. So I'm just going to give a quick example of a study that uses AL, activation likelihood estimation, because it's one of the most common methods used, and I want you to see different aspects of the methods just as a preview for some later lectures. So they found 704 studies and whittled that down to 11 studies to be included based on their inclusion criteria. Each study could contain multiple contrasts, so they're looking at the neural correlates of familiarity in music. So one contrast could be familiar music, looking at activation during familiar music as opposed to unfamiliar music. So here we see the results areas where activation was greater during familiar music compared to unfamiliar music. And another contrast they looked at was unfamiliar over familiar music. So here we see areas that were active during unfamiliar music as opposed to familiar. And I've included different details of what each contrast involved in the meta-analysis, just to give you an idea of what the data looks like once you're pooling studies together. So they had 10 studies in this contrast, but that involved 128 activation foci, so those are the coordinates of peak activation. And across the 10 studies, they had 196 participants. So again, they used AL with a software called GingerAL, and I'll mention that again next slide. But there are a few limitations. There are relatively few studies included, especially in this contrast. Some authors recommend using 17 studies as a minimum for AL, whereas some other authors recommend 30. So this is based on different simulations to see how well AL does with different numbers of studies included. Further, only 4 of the original studies were corrected for multiple comparisons, so there's probably a lot of noise that's being meta-analyzed here. And all the individual studies had small sample sizes, which is pretty common in neuroimaging, and this is why we want to do meta-analyses. But it's still a limitation if the original studies had small sample sizes, because their results will be noisier. So here I'm showing the most common methods, and like I was just saying, the method that was used in that study, AL, is the most common method. So here I read about 250 meta-analyses and annotated which methods they used, and this is the distribution for the use of different methods. And you can learn more about this and other aspects of meta-analyses on my poster. A little bit of self-promotion here. The poster is about a review of neuroimaging meta-analyses. So one of the reasons why AL is so popular is that there's a software that makes it really convenient to do AL, and it's called GingerAL. And one of the benefits of some new efforts like Nightmare, which we've talked about, is that it implements many different analyses so that you have more options and don't necessarily have to use the software that's available and easiest to use and have that decide which method you're using. Not to say GingerAL isn't great. It's a great software. You could use it if you wanted to. It's just we're trying to encourage people to think more about which method they're using and have more options for doing meta-analyses. So say you want to compare multiple groups of papers, there's different interpretations for these 2 methods, and actually it boils down to 3 methods because there's 2 different versions of the MKDA chi-squared test. So first of all, for scale, the interpretation is there more convergence in the sample being the sample of studies that's being meta-analyzed. So is there more convergence in the sample versus the null, defined as the base rate of activation in the literature? So that means across many different topics, there's this base rate of activation reported in studies, and they're using that as the null instead of, say, a uniform distribution. In MKDA chi-squared consistency test, the question is there more convergence in the sample versus chance? Whereas in the specificity test, they're looking at convergence in the sample versus an unrelated sample. So there you might have a sample of studies that look at music, and then say a sample of studies that are similar in ideally every respect except for they didn't have music. So maybe some other type of auditory stimuli or so on. Next, let's look at the branch for functional connectivity. Currently there's just this one method that we can use for this, and it's an approximative connectivity where connectivity is approximated as areas that are frequently active together in the literature. So basically you have an area of interest, which we'll call a seed, and that can be a mask or a coordinate, and that will make sense in the next slide. And then you want to see which areas are frequently active in the literature with that masked area or that coordinate. So here on the left we have an amygdala ROI mask, and here we have a spherical ROI, which is a coordinate just convolved with a sphere so that it's bigger. And here we see the meta-analytic co-activation maps for the ROI and for the sphere, and they're pretty similar. So of course there's a lot of connectivity with the amygdala itself, but here you can see other regions that are frequently active with the amygdala, and that's interpreted as a kind of connectivity. So as an example of a paper that uses this, in this paper they had different areas of the fusiform gyrus that they were interested in. These areas they had distinguished previously based on cytoarchitectonics. And they wanted to know which areas were they wanted to estimate the functional connectivity of these areas. So they did that using meta-analytic coactivation modeling. And these are the areas that were co-active in the literature with both of the areas, and these are areas in purple and green that were co-active respectively with the purple and green seed areas. And that's how we decide on a manual meta-analysis method in Nightmare. So next I'll talk about different methods you can use for an automated meta-analysis and when you might use them. So at the first stage, the question is, are you planning a study? And if yes, do you want to do encoding? So you don't have any results yet. You want to plan a study and maybe plan a region of interest where you want to look for results, or a seed region from which to look at connectivity. And there's another question to ask here is whether you want to do out-of-sample prediction or within-sample prediction, where the sample is the published literature. So if you want to do out-of-sample prediction, you'll be using a machine learning method. And these methods can take multiple words in order to predict what the results might be for things that actually haven't been examined as topics in the literature necessarily, or at least aren't one of the topics in, say, neurosynth, which has already been, the topics have already been modeled. So on the other hand, if you want to just summarize the findings within the literature, you would use a statistical method, but the limitation here is that the concept has to be one word. It has to be one of the words that are already in the vocabulary for a program like neurosynth, and I'll illustrate these two in a second. So here we have NeuroQuery, which is one of the machine learning models, and you can see I've entered a query that's more than just one word. I want to look at familiar versus unfamiliar music, and here you can see it's predicting some activation in the fusiform gyri. On the other hand, If I go to neurosynth, which uses a statistical method, a within sample method, if I have to choose music. I can't choose music. I can't choose unfamiliar or familiar music because it's not one of the topics that it contains. So I've chosen music, which is arguably the closest topic, and I've gone to the coordinates that we see here, and there is no indication from summarizing the literature that the fusiform gyrus is active during music. So it might be interesting to actually do a study where you use this as an ROI in order to investigate it. And just a reminder that this is actually the topic of that previous meta-analysis. So like neurosynth, they were summarizing the existing literature. They were doing within sample encoding, and they didn't have results in the fusiform gyrus, but maybe that's because previous studies were doing whole brain examinations and whole brain analyses and didn't have the power to find something in the fusiform gyrus. Who knows? So maybe that could be the next study you do. On the other hand, if you're not planning a study, the other stage where automatic methods can be useful is the stage of interpreting results. So this is where you have a map, you have some kind of result, and you want to decode that map. So you want to go from a map to topics instead of from topics to a map. So the first question here is whether that map is a continuous map or a discrete binary map, and which specific method you use is contingent on that, and I've given some recommendations here. And in other parts of the lecture, we'll talk a bit more about the dangers of decoding and interpreting decoding, because when you're going from a map to topics, it can be tempting to think, well, I got this map where the amygdala was active, and if I look in the literature, the amygdala is often active during, I don't know, looking at fearful faces. And then I might interpret, oh, this person must have been looking at fearful faces, but that's not necessarily the case. However, there is a study that did something like that. They wanted to decode the cognitive processes from the fMRI data of one individual. So they had one person sit through a number of different tasks, and those are named here. What was the sort of topic of interest in each task. And here we see the brain activation maps for the different tasks. Basically, they wanted to go from these maps to two topics in order to decode what the person was doing during each task. So they started with clustering the topic terms in neurosynth. So each of these dots is a topic term, and they've been clustered into these and spatially visualized in these different regions. And in the next slides, you're going to see this figure again, but where certain dots are bigger, and that means the map in neurosynth for that given dot, for that given topic, was related to the map of the individual person in the fMRI during certain tasks. So here we see their results from decoding with neurosynth, where they have different tasks listed above, and then they have highlighted by making the dots bigger and writing the words out of maps in neurosynth that were highly related to each task. So this is the kind of thing that you can do with decoding using something like neurosynth. So in conclusion, again, I want to emphasize this is a guide to get started. You don't have to understand every task. And even us teachers, we won't remember or understand everything all the time. the important thing is to know enough in order to get started with your own meta-analysis. And that's it for this lecture. I hope you've enjoyed it, and good luck with the rest of the course.